Good afternoon, ladies and gentlemen. This is a microbiology lecture, and it's about basics of medical immunology.
Good afternoon.
Just a minute. Basics of medical immunology. Okay, do you see the screen? Do you see information?
Yes,
do you see? Okay, so we will talk about immune system. The immune system is the complex of biological mechanisms of the body aimed at maintaining structural and functional homeostasis. That is, to protect the body from various non-communicable and infectious diseases. And the main function of body really is to keep homeostasis inside the body. The sensitivity of immune system is very high, and the immune cells are able to recognize the changes in one gene. So firstly, they check constantly on cells, and if they notice the changes in gene, the cell is destroyed. And of course, then the second function is to recognize microbes and different substances that enter human body. Elements of the immune system recognize there and align antigens and remove everything that is genetically different from it as a line. The organs of human immune system are divided into central and peripheral. Central organs are bone marrow and thymus. Peripheral organs are spleen, the lymph nodes, and accumulations of lymphoid tissue in the gastrointestinal mucosa, pair patches. Lymphocytes perform the main function of the immune system, highly specific recognition of foreign and altered self-antigens. Distinguish T lymphocytes and B lymphocytes. T lymphocytes that mature in the thymus and B lymphocytes that mature in the bone marrow or lymphoid tissue of intestine. They are differ by functions. T lymphocytes functions. T lymphocytes recognize antigens, provide the realization of the cellular immune response, and interact with B lymphocytes. They are after recognition of antigen, then they send special substances, interleukins, and finally it is long chain immune response, interleukin 1, and then another cells are activated, they will produce interleukin 2, and finally it goes to B lymphocytes and they are activated. B lymphocytes are precursors of antibody-forming plasma cells, and some of them are B memory cells. And plasma cells are cells, those are able to produce antibodies. Immune reactions are divided into humoral, occur with the participation of antibodies circulating in the body, mainly in the serum of blood. So humoral immunity and cellular. Cellular immunity occur by direct contact of immunocompetent cells. Both these immune reactions are important, humoral and cellular. Antigens, what are they? Antigens are substances of different origin recognized by the human immune system and causing immune reactions in the form of antibodies production, the appearance of sensitized lymphocytes, or the production of immunological tolerance. There are different kinds of immune response, maybe antibody production, maybe appearance of sensitized lymphocytes, memory cells, or the production of immunological tolerance. In case of tolerance, immune system is not able to react. So antigens properties like tolerance is developed for this own cells. When the immune system is developed inside the fetus during the pregnancy, all substances those the immune system is communicate to before during the development, immune system did memorize like own, and tolerance is developed. Okay, antigens properties: immunogenicity, antigenicity, and specificity. Most pathogens of human infectious diseases and their toxins are antigens that cause the development of immune response. Of course, they consist of different substances, and they are foreign substances for immune system, and they are recognized by immune system. Bacterial antigens, or antigen, just in general, every substance or bacterial cell is recognized like antigen, specific antigen, and specific antibodies are produced for every substance. But in practice, in science, of course, it's important to detect antibodies for certain substance, especially if you are going to produce subunit of vaccine. It is important. But in practice, we are not going to detect antibodies for every substance or bacteria. And basically, we are talking about O antigen, somatic substances of the cell wall, K antigen, capsular. Most of bacteria have capsule, micro, macro, and H antigen, antigen of flagella, protein of flagella. In case of bacteria have got flagella. If bacterial cell has no flagella, no H antigen. And exotoxin, if bacteria cell is able to produce exotoxin, it is a protein and it has specific antigenic structure. Of course, it is antigen and it is recognized by immune system. And specific antibodies are produced for O antigen, K antigen, H, and for exotoxin. Viruses consist of different substances, core antigens inside nucleic acid and internal proteins. Capsid consists of different proteins. They differ, and they differ by antigenic structure. And in envelope, there are hemagglutinins and neuraminidase. There are spikes. Really, in envelope, there are no viral substances. There are mainly lipids from the host cell, but spikes go through the envelope and they are on the surface, and between them there are hemagglutinins and neuraminidase, and they differ by antigenic structure. Other microbes, such as fungi and protozoa, of course, consist of different substances, and fungi and protozoa are recognized, of course, like antigens, but we are not going to detect antibodies for certain antigens. Anyway, in case of mycosis or protozoal infections, antibodies, specific antibodies are produced for every causative agent, and these antibodies are detected in general just for some causative agent. Prions consist of just proteins, and they do not cause immune responses because of they have similar antigenic structure with proteins. Those are produced in the head brain. So, and they do not cause immune response, unfortunately. So diseases caused by prions are non-treatable, and immune response is not developed. Antibodies, what are they? Antibodies. Antibodies are effector molecules of humoral immunity. Synonyms are glycoproteins, gamma globulins, immunoglobulins. These terms are used for antibodies. So they are glycoprotein, they are gamma globulins, and they are immunoglobulins. Distinguish serum antibodies. They are circulated in the serum of blood, and so serum of blood is used for detection of serum antibodies, very important in diagnosis of infectious diseases. And after this word, the method for detection of antibodies or antigens is named serological method. So serum antibodies are detected for proving of infectious diseases. Doesn't matter bacterial or viral or protozoal or mycosis. And there are secretory antibodies. They are in biological fluids, in saliva, tear fluid, or mucosa membranes. They are important, of course, because they are developed immediately after the entering of pathogenic microbes. But in diagnosis of infectious diseases, we are not going to detect them. Very rarely, sometimes such liquid fluids are used. Cerebrospinal fluid, maybe saliva, very rarely, okay? Very often we are looking for serum antibodies. Those are circulated in the blood. Immunoglobulins are divided on different classes. There are classes of antibodies such as IgM, IgG, IgA, IgE, IgD immunoglobulins, and they are looking differently. I will show the picture classes of immunoglobulins, and this is just scheme. This is subunit of immunoglobulin. It consists of light chains and heavy chains, and immunoglobulin G consists of one subunit, the same like immunoglobulins E, immunoglobulins D. Immunoglobulins A consists of two subunits, and immunoglobulins M consists of 5 subunits joined in one. Okay, and okay, so and they are differ by functions. Immunoglobulins M, they are primary antibodies. They are synthesized by immune system when the antigen enters the body. Detection of immunoglobulins M in the serum of blood indicates about fresh infection, just the initial stage of infectious diseases. And but they are not, immunoglobulins have lifetime. They're not circulated in blood constantly. And immunoglobulins M are produced, and they are able to react specifically for antigen for those they are produced with developing complex antibodies, antigens. And if antigens are not deleted from the human body, microbes are not eliminated, immune system still produce antibodies, another class. Immunoglobulins G, the main class of antibodies. 75% of all antibodies, those are circulated in blood, are exactly immunoglobulins G. They are detected in healthy people just in very small amount in the blood. And immunoglobulins M, very, very rarely, if immunoglobulins M are detected in the serum of blood, means the person was infected recently by some microbes, and the task is to find exactly what microbes, what qualitative agent this person is infected by. So they produce later during infection, and they are detected in the chronic stage sometimes. So they indicate about the last stage of infection. And they are named, they are protective antibodies. They are able to protect. Immunoglobulins A, they are present in saliva, tear fluid, milk, breast milk, on the surface of mucosa membranes, and in a small amount in the blood. Immunoglobulins E are involved in allergic reactions and not caused by microbes. Microbes are caused hyperdilated sensitivity, cell-mediated. So for detection of allergic reaction, we are not going to detect immunoglobulins E. Microbes don't cause production of them. We are going to detect hyperdelayed sensitivity, cell-mediated. And immunoglobulins D are detected, biological role not established well, definitely. Then classes and types of immunity. Local immunity protect the skin and mucosa membranes from pathogenic effects. The main mechanisms are secretory antibodies, mainly immunoglobulins A, and phagocytes. They participated in local immunity. General immunity provides generalized protection of the internal environment of the body from pathogenic influences. And species-specific immunity, genetically fixed immunity inherent in each species. For example, human beings belong to species Homo sapiens, and we have species immunity to some pathogens of plants. For example, we are resistant to pathogens of plants. They have viral infections, bacterial infections, they have mycosis. but we have immunity, species immunity, and animals have own pathogens, but mainly human beings are resistant to animals' pathogens. We have species immunity, and it is proved that human beings have species immunity to bacteria of Mycobacterium genus, to mycobacteria. But anyway, mycobacteria are able to cause tuberculosis, in in human beings, and leprosy, Mycobacterium lepra, and other species are able to cause so-named atypical mycobacteriosis. And but realize many people are infected by mycobacteria, but clinical symptoms are not developed. Infectious disease is not developed in spite of people are infected because of species immunity. And the only immunocompromised people, those have immune deficiency from by nature, from the birthday, and maybe they have acquired immunodeficiency. They just on this background of immunodeficiency, any kind of immunodeficiency, mycobacteria are able to multiply, and clinical symptoms of tuberculosis or leprosy will be developed. So it is a sign of immunodeficiency. Then acquired immunity. We are talking about acquired immunity, kind of acquired immunity. They can be natural, active and passive, or artificial, again active and passive. So naturally acquired active immunity develops after an infectious diseases or after the hidden contacts with microbes, so-called household immunization. In this case, if microbes enter human body, they are recognized by immune system, and after recovery, the patient will get acquired active immunity. In case of active immunity, immune system should be activated. Natural immunity is developed due to natural infectious diseases, and natural immunity can be lifelong after measles, for example, long-term after typhoid fever, for example, or relatively short-term after diphtheria, natural immunity. Sometimes immunity is a very narrow, like type-specific immunity, like in case of influenza, for example, because of strains of influenza differ by antigenic structure, and immunity in case of influenza, for example, is not species immunity. Patient has immunity just for certain serological antigenic variant of influenza virus. Anyway, immunity is developed, but it's not species immunity. Okay, then naturally acquired passive immunity develops through ready-made maternal antibodies transmitted to the fetus through the placenta and then to the newborn through the breast milk. It is natural, and but it is a quite passive immunity because of the in passive immunity, the fetus and the newborn will get ready antibodies from the mother's body, and the immune system is still passive. It's not activated, okay? And antibodies are not able to circulate during the life of this newborn. They have lifetime, and just during a few months of life, the first months of life, newborn is protected by action of maternal antibodies, and then they are destroyed. So, and then the natural maybe infection when immune system is activated, and maybe passive immunity when the newborn gets ready antibodies. What about artificial acquired immunity? Artificially acquired immunity, again, may be active and may be passive. So artificially acquired active immunity develops after immunization with vaccines. In this case, patient is not infected by natural way. He is vaccinated. Vaccines are prepared from microbes. maybe live microbes, maybe inactivated microbes, or maybe some individual microbial substances, just substances of microbes. Anyway, vaccine, by using of vaccine, microbes enter human body, microbes, or maybe just some certain particles of them. And the immune system should be activated. Again, the immune system should recognize these antigens, and immune response should be developed by using of antibodies production, memory cells production. So, and vaccination gives immunity, artificial immunity. The body is actively involved in creating immunity, responding with the development and of an immune response and formation of memory cells. But vaccination doesn't give, again, immunity for the whole life. So newborns are very sensitive to infectious diseases, and newborns are vaccinated during the first year of life very often by different vaccines. And in certain intervals, we should give them booster doses. Again, the vaccination should be done again to activate immune system. So, and we are going to protect children during the first years of life. But after that, the vaccinations are done in accordance with national vaccination schedule. In all countries, there is the specific national vaccination schedule, but many vaccinations are used all over the world because diseases are common in some countries, like whooping cough, like measles, mumps, and rubella, like diphtheria and tetanus. And these vaccinations are necessarily all over the world for all children. And sometimes they are continued for an adults, like vaccination against diphtheria and tetanus is in Russia, for example, by Russian national vaccination schedule. An adult should get this vaccination every 10 years, 16 years, and 26, 36, 46. For the diphtheria and tetanus are still dangerous for an adults. But realize that after again, this immunity developed due to vaccination is not life-lasting. And finally, this child that was vaccinated during the childhood, finally, he has no artificial immunity due to vaccination, and he has no natural immunity because of he had no measles, mumps, and poliomyelitis in childhood. So, an adults are not protected, and an adults are able to be infected by the measles virus, mumps virus, by causative effects of whooping cough. So it's not just children's infectious diseases, and the adults have the same diseases because of no, they're not protected. Artificial immunity is not life-lasting immunity, of course. Then, what does it mean, artificially acquired passive immunity? In this case, it is achieved by the introduction of ready-made antibodies. Preparations containing antibodies are named immune sera and immunoglobulins. So in this case, patient will get injection of preparations. Those consist of ready antibodies, and again, immune system still passive. Just get ready antibodies, and ready antibodies are again, they are not able to circulate during all life of patient. The immune system, but in this case, the immune system reacts passively, not participating in the development of immune response. So, vaccines for creation of artificial active immunity, vaccines are used. What are they? Vaccines are immunobiological preparations designed to create active specific immunity. They are used for specific prophylaxis of infectious diseases. The active principle of the vaccine is a specific antigen. All vaccines consist of antigens, and because of we are going to stimulate immune system. What are they? antigens. As antigen are used live microbes, but with mild virulence. Virulence is decreased, and for vaccination, special vaccine strains are used. The first vaccine was produced by Louis Pasteur, and he was the first microbiologist who did realize that it is possible to decrease the virulence of microbes. He did cultivate positive agent of anthrax in unfavorable conditions, and finally he did get strain of Bacillus anthracis, and these microbes, they're still alive, but they were not able to cause anthrax. And after injection of these microbes, firstly, he did use on animals, he did prove on animals that it is possible to protect the animal's body from microbes by using of vaccines. He did give name this vaccine for this preparation, and he did infect these animals by viral strain on Bacillus anthracis, and infectious disease was not developed. So he did prove that it is possible to use microbes for vaccination. And after him, vaccine against tuberculosis was produced by Bordet and by Calmette and Guerin. They did develop BCG vaccine, and BCG vaccine abbreviation means Calmette and Guerin bacillus. They did cultivate the strain of Mycobacterium bovis during 13 years, 13 years, and finally they did decrease the virulence of this strain, and now this vaccine strain is used for vaccine production. This vaccine is used in many countries, not in all countries, unfortunately. In our country, the BCG vaccine is used on the age of 3 days of life at maternity hospital. Newborns are vaccinated. So, and after that, other vaccines were prepared from live microbes, like measles vaccine, mumps vaccine, rubella vaccine. They are live vaccines. And then Sabin did produce a live vaccine for prevention of poliomyelitis and so on. And attenuated vaccines are used till now for prevention, for prophylaxis of many infectious diseases, bacterial and viral. And killed microbes are used, and vaccines, those consist of killed microbes, are named inactivated vaccine. They are not alive, these microbes, but they are not destroyed completely. They are killed very gently by using of ultrasound, for example, because the antigenic structure should, we are not going to destroy antigenic structure, killed microbes. And Salk vaccine, again against poliomyelitis, is used, a kind of killed vaccine. And now killed vaccine is used for prophylaxis of rabies. Against whooping cough, killed bacteria were used in DTP vaccine, so on. So killed microbes are used for vaccination, the same. Viral vaccines. And sometimes, so I'm repeating, in vaccines as antigen are used live microbes, the whole microbes, whole bacterial cell, the whole viruses, maybe killed microbes, again, the whole bacteria, the whole completely virus particles, but some substances of them are toxic. And to avoid harmful effect, now vaccines are prepared just from some particles of microbes, so named so-called protective antigens, isolated from microbes. Because of, unfortunately, sometimes side effects and complications are developed after vaccination. It's not good, of course. And so the new generation of vaccine now consists of just specific protective antigens. The task is very important for pharmaceutical faculty students' production of vaccines. The task is for scientists to destroy the some certain microbes and to isolate 13 substances of these bacteria or viruses. And every substance should be used, of course, on laboratory animals, like vaccine, and then we should realize exactly which one substance is able to cause production of protective antibodies. Those will protect this body from the whole microbes. And polysaccharide of meningococci was detected like protective antigen, and it is used for vaccination against disease caused by meningococci. And hepatitis B antigen, surface antigen, HBs antigen was detected like protective, and it is used now for vaccination against hepatitis B. And like antigen, sometimes exotoxins are used in case of toxinemic infections. We are going to create antitoxic immunity, and exotoxins are isolated and they are treated by formaldehyde during 37 days at temperature 38 degrees by Celsius. And finally, we will remove toxic properties, but it's still the same proteins, and preparations prepared of them are named toxoids, anatoxins. Toxoid in English, anatoxin in Latin, and they are used for specific prophylaxis of toxinemic infections. And sometimes chemically synthesized antigens are used, not often. It is just idea, but they are not produced now yet. And then antigens received by using of method of genetic engineering, recombinant vaccines, and the vaccine against hepatitis B now is kind of recombinant vaccine. Then on laboratory class, I will explain you what does it mean. So vaccination is an injection of weakened form of active antigen, and so on, so on. So vaccination is a very useful procedure, okay? So serum immune preparations. Serum immune preparations are named immune sera and immunoglobulins. Immune sera. Immune sera are obtained from the blood of immunized animals or the blood of immunized people. Purified and concentrated immune sera are called immunoglobulins. So just immunoglobulins are isolated from immune sera, no other proteins. Immune serum preparations obtained from the blood of animals are called heterologous, and the blood of people homologous serums. The activity of serum preparations is expressed in antibody titers. Serum immune preparations are used for treatment and for diagnosis. So specific treatment and emergency prophylaxis of infectious disease is used for treatment of human beings, of course. Healthy people don't need ready antibodies, so just patients and people who was in contact. And there are diagnostic immune sera. They are used for diagnosis of infectious diseases to detect microbial antigens by using of immune tests. Immune sera and immunoglobulins are used to treat diseases. Of course, it is better to use homological sera for treatment. Unfortunately, we have no donors enough. Donors should be vaccinated. Donors are people who give the blood for medical purposes, but they should be agreed to be vaccinated. Sometimes they are not agreed, so unfortunately, Hosi serum is used. Not good. Immune sera containing antibodies to microbes are called antimicrobial, antibacterial, antiviral, and containing antibodies to bacterial exotoxins are named antitoxic serum. Sometimes drugs containing antibodies are used for emergency prophylaxis in case of suspected human infection without clinical signs of the disease for people who was in contact with the patient and they are infected for sure. For example, tetanus antitoxic serum when the patient has some wound. It should be used for any case rabies immunoglobulin after an attack of an animal with suspected rabies. Passively acquired immunity doesn't last long, of course, and disappears as the donor antibodies are removed from the bloodstream. So not necessarily for healthy people. Medicines are represented by a foreign protein after the introduction of which adverse reaction often develop. I told you, unfortunately, Hosi serum is used. Even the human beings' immunoglobulin, sometimes reactions are developed. So thank you for your attention. Have you any questions? Have you any questions about the lecture? It's very important topic, and then we will discuss, of course, on laboratory classes.
Okay, thank you, doctor.
Of course, and we will study immunology during 2 laboratory classes. I'm not able in 40 minutes to explain you all immunology. So my advice is you should read the textbook. There is immunology part in any microbiology textbook. So my advice is you should read microbiology textbook in your native language. Of course, we are not going to study completely immunology. I've sent you the list of questions for test number 3. So please find the information in accordance with list of questions and be ready for class. And on class again, we will discuss all these questions. And it's very important because of you have to know all these preparations. Those are used in medicine, in diagnosis of infectious diseases. And you should know vaccines. What are they? All types of vaccines. You should know in examples. Really, you should learn by heart all of them, your pharmaceutical faculty students. And then we have to know preparations. Those consist of antibodies, immune sera, and the serum is singular, immune sera is plural. So preparation is named serum. Really, preparations are named in short just serum, very short name, but by meaning, what does it mean? The full name should be hyperimmune serum of blood, but preparations are named just serum in short. And some sera are used for treatment. They are very useful. Unfortunately, sometimes horses serum is used for treatment of diphtheria, for treatment of tetanus, because the diphtheria and tetanus are very common infections. And we have no donors enough. Those are ready to be immunized by toxoids. And so it is possible to use horses. Horses are immunized by using of any vaccine or maybe toxoid. It depends what antibodies do we need. In case of diphtheria and tetanus, horses are immunized by toxoids, tetanus toxoid and diphtheria toxoid, and we will get antitoxic serum. And horses serum is used for treatment and for emergency prevention in case of tetanus. Of course, they are foreign. Horses serum consists of antibodies for exotoxins of causative agents of diphtheria or tetanus, but on another hand, they are horses proteins. And if horses proteins enter human body, they are recognized like foreign antigens by immune system. And sometimes allergic reactions are developed. So before using of horses serum, the doctor should do skin allergic test. Always there is one ampoule with diluted serum, and it should be injected into the skin of patient, and we have to wait 30 minutes to see. Maybe it's not the first time of injection of horses serum, and maybe patient has delayed hypersensitivity. So we should check always. Of course, it is better to use homological serum. And if donors are immunized, after we will get hyperimmune serum of blood by using of chemical method, we should isolate just globulins, because antibodies do not belong, not all proteins are antibodies. There are many proteins in the serum of blood of human beings, but not all of them are antibodies, just globulins. So firstly, we should isolate by chemical method just globulins. And again, not all globulins are antibodies. They should be divided on fractions, and just gamma fraction consists of antibodies. And purified preparation is named gamma globulin. Now, another name is just immunoglobulin. It is concentrated and purified preparation of human's origin. Nowadays, sometimes horses immunoglobulins are used. So if you have no any questions, now the time is over and I will finish the lecture. Okay, so and keep your questions and we will discuss on laboratory classes and other teachers will explain you. But my advice is you have to read the immunology part in any microbiology textbook. Okay, so have you any questions?
No, doctor.
No questions. So now all students are present, all groups students. Finally,